Hello and welcome to part 2 of the tutorial for Lab 8. Hopefully you have already watched the overview and part 1. Alright, I'm just going to go into the lab, and again you should have the instruction notes handy and you should have the simulation data worksheet handy. Remember, the simulation data worksheet looks like this. You can fill it in by hand as you're running the experiment and then transfer it over to an Excel spreadsheet after. Now, when you look at the actual lab instructions, you'll see that it says you will run a simulation and study a population. You're going to change two primary characteristics or settings. You're going to change the mutation rate, which is how fast these particular organisms are disturbed, how fast they mutate or change. Okay. You are then going to change the selection rate. The selection rate is related to how those organisms interact with the environment. So the greater the selection rate, the greater the interaction, and that means that ideally you're going to see more of the influence of the environment on the organisms. Now, what happens with the organisms, if we go to the evolution lab, and remember you get there by going to the biology in motion, is that we're going to take a population. We're going to take a population of these blue grabbers. And their ideal is they have to capture this prey, and by capturing the prey, they will end up being able to stay around and reproduce. If they do not have food, they have the potential to die. Because what happens is you will have an organism that dies and then an organism that reproduces to fill that empty space. The organisms that have not eaten are the ones that have potential to die. The ones that are eating the most and are the healthiest are going to be the ones that have the best potential to reproduce. And in this world, when they reproduce, ideally they reproduce the same organism. Meaning, if their grabber is so big, then they reproduce with an organism that looks like them. Of course, if there's mutation, that changes. And the only thing we're going to mutate here is this piece. So when you go to the simulation, what you're going to do is you're going to watch the population down here, and you're going to change the settings up here. And then you're going to go through what are called cycles. Cycles are reproductive cycles. During a cycle, you'll have food come down. One lucky grabber gets the food, and that increases their health. Then all of the grabbers are compared. Whichever one has not been eating, is most unhealthy, dies. Then the grabber that is most healthy reproduces and fills that empty spot. If there's mutation, then the grabber may look different. All grabbers start with 2. That's what mean phenotype means. It's how many of these grabber sections does your population have. Mean is kind of like an average. What's the average size? So what you're going to do is you're going to run this simulation. You're going to look at how these change or don't change and record your data. In the next part, I will go over specifically what you're going to change and specifically what you're going to record.